Today we'll be looking at single nucleic acid point mutations, and there's 4 different types: missense, nonsense, silent, and read-through. Missense is a mutation that results in a different amino acid being produced. A nonsense mutation results in a change in a codon, so it becomes a stop codon. A silent mutation results in a same amino acid being produced. And a read-through is when a stop codon is changed to a codon that codes for an amino acid. There's 2 different types of these types of mutations that can occur. So there's a transition, which is a purine turns to a pyridine, or pyrimidine turns to a pyrimidine. So adenine turns into a guanine, or a cytosine turns into a thymine. Or there's a transversion, which is a purine to a pyrimidine, or a pyrimidine to a purine. So an adenine turns into a cytosine, or a cytosine turns into an adenine. So this is our wild type DNA. So there's no mutations in this coding strand. And so from 5' to 3' is our coding strand, and below that is our mRNA sequence, and below that are the amino acids that will be coded by our mRNA sequence. There are no mutations, so this is what we're supposed to be getting. And so we're going to compare these amino acids to the next couple of examples to see what type of mutation we have on our DNA strand. So this is a missense mutation, and the top strand is the wild type, and below that is the mutated strand. This is a transversion. As we can see, the cytosine turns into an adenine, so a pyrimidine turns into a purine. This mutation results in AGA being formed as a codon in the mRNA sequence, and this will result in arginine being formed instead of serine as we had in the wild type. Our next example is a nonsense mutation, and as we can see, the adenine is going to turn into a thymine, so it's a transition from purine to pyrimidine. So now this mutated strand will be coded for mRNA, and we can see that UAG is now formed, and this will produce a stop codon instead of lysine produced in the wild type phenotype. Now we're going to look at a silent mutation, and as we can see in the wild-type strand, the codon is AGC, but in the mutated strand it turns to AGT, and C to T is pyrimidine to pyrimidine, so it's a transition. So for the mRNA sequence, this is going to produce AGU instead of AGC, but a silent mutation has no effect on the amino acid produced, so both AGU and AGC both produce serine. Lastly, we're going to look at a read-through mutation, and as we can see, the codon TAA is turned into an AAA, and so this is a transversion, turning a pyrimidine into a purine. So this will result in a new mRNA strand, AAA in the mRNA, which will produce lysine. Before we had UAA, which was a stop codon, and so we're having a stop codon turned into a codon that produces lysine. So this is a read-through mutation.